I'm so excited for summer. How about you all? One of my favorite things to do on a warm, sunny day is to lay in the grass and look up at the clouds. With a little imagination, those lumpy, wispy clouds take on the shapes of animals, scenes from nature, whatever comes to mind. But what shape are the clouds really? From your vantage point on the ground, you can't see how high in the sky the clouds extend. You also can't see that the clouds are actually made up of tiny water droplets that control the shape of the clouds and the probability of rain. As a scientist, when I think about clouds, I can't help but notice a similarity between clouds and tumors. Tumors, like clouds, have lumpy, irregular shapes with tendrils that extend into the surrounding area. They're made up of smaller parts, cells that control how they grow and behave. In fact, recent research has shown that looking at the behavior of cells can help to predict when a tumor is likely to metastasize or spread, just like how looking at the size of water droplets in a cloud can help to predict when it will rain. In order to fully understand a tumor or a cloud, we need to be able to see them on two different scales of size. We need to see them on a big scale to visualize the overall shape, but we also need to be able to see them on a small scale to look at the component parts and see how they influence the overall behavior. My research is all about developing tools that allow biologists to better study how the behavior of cells influences tumor growth. Using fiber optics, I'm developing miniaturized versions of different types of microscopes that fit within a standard hypodermic needle. Doctors and biologists can insert this needle into their sample to image from the inside to enable better detection of tumor boundaries or to study the impact of the arrangement of different cell types within a tumor. If the standard microscopes that biologists currently use are like looking at the clouds while standing on the ground, these new tools I'm developing are like drones that could fly up into the clouds and image them from the inside. Then, from a better vantage point, you can more easily map out the shape of the clouds and maybe even look at the size and shape of water droplets in different areas. But there's a trade-off. These tools need to be able to image on a small scale over a large volume or field of view, and they also need to be small to minimize the damage to the sample from inserting the needle. And it's a challenge to develop tools that satisfy all three of these criteria. So I'm investigating the design of novel lenses that can be fabricated on the end of optical fibers that will shape the light into non-traditional shapes to overcome this limitation. This will allow biologists to see more cells at once to enable the study of how interactions between cells influence tumor growth. The hope is that one day we'll be able to classify tumors as easily as we can classify clouds and predict metastasis as easily as we predict the rain. Thank you.